Hello everyone, welcome to today's lecture. Just before we get started, I just want to remind all of you, we do have a class test next week Tuesday, so keep in mind we will not be having a lecture then. The exam or class test will be taking place during that time period, during the scheduled class period. So for that reason, we will not be having class. The exam will be available during that class time period, the scheduled class time period. But you will only have 1 hour to complete the exam within that time period. So please schedule your other classes and activities accordingly. It's nothing too serious. All as we discussed before, and as I've informed you, this will all constitute short questions, so multiple choice, column A, column B, true or false, kind of similar to the examples we've been that I've been showing you guys at the end of the lectures, barring obviously long questions. This class test will not have any long questions. Okay, guys. So as I said, today we're going to be talking a bit about the control of microbial growth. This builds on our lecture of last time where we looked at microbial growth. So now we're going to be discussing ways in which we can control microbes. So why is this important? So controlling microbial growth is important for obviously in the medical field, as we're all aware, with COVID being a thing, and then also in pharmaceutical and biotechnology industries. It can also be used in academic research. Think back to that long-term E. coli experiment, and in the food industry. So there are numerous methods that we can use to control microbial growth, and some of these are on our slide here. You will see that there are some terms that I definitely want us to just go over here quickly, because these terms you'll have to know going forward to make sure you understand the work. So first off, starting on sterilization. So what is sterilization? So sterilization is any process that eliminates or kills all forms of microbial life present on a surface, a solution, or on solids. Generally, this will involve extreme physical or chemical processes such as heat, chemical irradiation, high pressure, and filtration, or it can even take a combination of those approaches, if you really want to be sure. Think of running, sticking a surgical or a slide over a flame or something like that, or an instrument over flame to just make sure that you kill all the microbes. Your inoculation loop in the lab is a good example, where you will run it through a flame to make sure that it is sterile. Going on to an antimicrobial agent over here, that's a general term for drugs, chemicals, or other substances that either kill or slow the growth of microbes. Note that they can kill or slow the growth. Now we're moving on to a disinfectant. So what is a disinfectant? So unlike sterilization, a disinfectant doesn't necessarily kill microorganisms, and especially not resistant forms of microbes where they form spores with those tough outer shells. So disinfectants work by destroying the cell wall of microbes or interfering with their metabolism. And then what is an antiseptic? An antiseptic is an antimicrobial substance that kills or inhibits growth, and they are applied to living tissue or skin to reduce the possibility of infection or sepsis. So what's the difference then between an antiseptic and a disinfectant? Well, the difference is that a disinfectant is only applied to non-living objects, whereas an antiseptic is only applied to living tissues. That's the difference in the terminology there. So then when we discuss antimicrobial agents, we'll also be using the terms bacteriostatic or bacteriostasis, a microbiocide, or to talk about controlling these organisms. So what's the difference there? As you might guess from the name, the stasis implies stopping something. So bacteriostasis inhibits the growth of bacteria without destroying them. They don't kill the microbes. So they will also not really impact the viability of your organisms, not necessarily. And then a microbiocide, these are treatments that are used to kill microbes. That suffix, the side over there, implies it means to kill. And stasis means equilibrium, or just think of something being static, not moving. Just you're interrupting their growth. Okay. So there are different approaches and targets on how microbial growth is controlled. First, there is general coagulation or oxidation, where coagulation refers to protein denaturation, in which proteins revert to a two-dimensional structure and become non-functional. This can be achieved, for example, by boiling the substance that the microbes are in, like water, for example. And then oxidation is the process that removes electrons from molecules. We'll be discussing more on that in the next lecture. So just be aware of the term. And this means that when you're taking away electrons, it will disrupt the cellular enzyme and the cellular structure of the bacteria. And if you disrupt the cell wall, you can affect important enzymes and DNA. So a good example here would be chlorine in water that's used as an oxidative agent that's used to kill the bacteria. Then we can also have bacterial agents that are more specific to certain microorganisms that cause damage to either the cell walls or the cell membranes, and that can interfere with specific metabolic processes. For example, you can target only gram-positive bacteria or only gram-negative bacteria. I will be discussing a bit more of that later in the lecture. And then we're also going to be looking at the bacterial death pattern. So that's the rate at which bacteria dies. And this is fairly consistent. And it usually occurs at an exponential rate or a decay, what's called the decay curve. This is an important one because we want to understand, what agents are needed and what their doses should be to actually control the microbial growth. And that's why we need to understand this death rate, how quickly we are able to eliminate the organisms or reduce their numbers at least, or stop their growth effectively. So yeah, it is also then obviously important for the development of standard protocols and determining the minimum time needed to achieve acceptable levels of sterilization and disinfection. So what we have here is an example of microbial death rate. So as you can see here, for every minute of time that passes, the death rate decreases by a factor of 10. So that means if you start with 1 million organisms, after 1 minute, 90% of the population has died. So now we are left with 100,000 microbes. And this then continues on and on and on and on. So for each minute that the treatment is applied, in other words, 90% of the remaining population is being killed. And that's why you have this death curve that's also typically displayed on a logarithmic scale. So here you can see on the y-axis here, we have the log scale, and that's represented by the red line. And here you have what's on not a logarithmic scale. This is just a normal scale, and that's the dotted line. See how much easier it is to just work on the logarithmic scale because it provides you with this nice straight line, which you can then use to calculate death rate and rate of disinfection. And so it's much easier to calculate the rate of change by using the slope here than this ever changing line that we have here. So what are the factors that can influence the success of antimicrobial agents? So there are actually quite a few of these, and they're listed over here. So we'll be discussing these now. This can be the concentration of microbial agent, concentration of microbes, time of contact temperature, kind of microbes. Obviously, different species will have variability, physiological age of the microbe, physiological condition of the microbe. So is it in its spore state or not, for example? And then the nature of the material bearing the microbe. So you have to keep in mind that depending on what you use, every microbial agent has optimal conditions under which it can function. For example, why do we only use a 70% ethanol to disinfect surfaces? That's because if you were to use 100% constant ethanol, it would harden the protein wall around microorganisms. So instead of the ethanol penetrating the membrane, the microorganisms will just go into dormancy. So if you want to kill the microbes or effectively suppress them, you have to know what the specifics of your control agents are and how you are supposed to effectively apply them. So that obviously then also means in the case of ethanol that more is not better. At 70%, protein coagulation will occur much slower and allow the ethanol to penetrate and denature the lipids and the proteins within the cell before that hardened outer shell can form. Then some of the more the other features here, more microbes are obviously harder to kill, especially if you look at something like biofilm formation. This is a big industry. You know, a lot of industries, if you look at oil pipelines, look at how to remove biofilms. It's big in the medical industry. So plaque is a form of a biofilm, because biofilms make it much harder for the antimicrobial agent to penetrate, because of the consortium of microbes essentially produce an extracellular polymetric matrix. And then time is another consideration, because longer time is generally better, which is why understanding the death rate is so important. You don't want to tell someone something has been disinfected, but then you didn't disinfect it long enough, and then there's potential for contamination or illness or whatever the case might be. So warm temperatures also tend to increase the efficacy of disinfectants, as with many chemical processes. Remember, more increase in temperature means more molecule movement, so you'll have a greater efficacy, more chemical reactions taking place. And then I'm going to touch briefly on the types of microbes, where some microbes are being more sensitive while others are more resistant. Thus, you have to identify your microbe. You have to know what you're working with and what you are trying to guard against or protect against. So moving on to the effectiveness of antimicrobial agents that we just discussed, just giving you guys some more details here. As we said here, this graph represented on the y-axis, we have the logarithmic number of microbial survivors and then the number of time. As you can see here, a higher initial population will take longer to get rid of. That's why your initial bacterial concentration is so important. If you start with a small concentration, it'll be much easier to decimate than if you start with a high concentration of microbes. And then you also have to keep in mind the environment that you're working in and where these organisms are adapted to. So under certain environmental conditions, antimicrobial agents can be inhibited. For example, biofilms, as we discussed before quickly, can interfere with disinfectants and make it harder to reach the target organism. And a lot of these, like I said, just as a reminder, an environmental condition that is really important is something like temperature, with warmer temperatures typically elevating the effectiveness of your disinfectants. And based on this graph, you can also see that time is an important factor, because if you had this starting high concentration here and you only ran it for 3 minutes, because that is what worked in a previous instance, for example, you would not have a total removal of your microbe. Where if you run it for long enough and you increase your time period, your time of disinfection or antimicrobial treatment, it will be more effective, of course. So discussing a bit more here about temperature, we're going to be talking something that you guys might have heard in the lab that Amy and Kieran discussed, something like autoclaving, boiling, pasteurization. Think back to Louis Pasteur. So that this is all forms of moist heat that you can use. Then you also have dry heat, which is used in ovens or incineration and results in a decimal reduction time. So with moist heat kills microorganisms through also through the coagulation of proteins. Let me just grab a sip of my drink here. Okay, meaning they also disrupt the three-dimensional structure of the protein, and the effectiveness of this method varies depending on the method used. So autoclaving uses high pressure and steam and is much more effective at killing microbes, including endospores, than boiling it, for example, because you have that double effectiveness. You have the pressure and the steam, not just heat. So just another note here, what is this Gentilization? Gentilization is a simple and effective sterilization method that is used today in substances that need to be sterilized, where the substance is heated to 121 degrees Celsius for 15 minutes in a pressurized system. And then, as I said before, when we are using high temperatures, we need to take we need to take into consideration the decimal reduction time, which is the time that is required at a given temperature to kill 90%. Remember, we want that 90% reduction. So this is this decimal reduction time is the amount of time it takes to kill 90% of the exposed microorganisms. And this is obviously then related to bacterial heat resistance. Certain archaea, if you think of your extremophiles, will be more resistant to heat than just your normal mesophiles. So then on the other side, you can also use low temperatures, think refrigeration or freezing. And this obviously depends on the temperature and the intensity, but low temperatures between 0 to 7 degrees Celsius can typically prevent microbial growth. And so they kind of mimic this bacteriostatic effect. And then if you freeze something below -2 degrees Celsius, this is more harmful because ice crystals can form and they can disrupt the cellular and molecular structure of the microbe. Looking at drying and desiccation. So as the name implies, pretty straightforward. This is where you just remove water from the microbe. This is harmful to organisms as anything else that's alive. The microbes need water to grow and reproduce. So this method has been used for centuries to preserve food. Radiation, obviously something a bit newer, or that we're only grown to understand in the last couple of centuries. So depending on the wavelength, intensity, and duration, radiation can have various effects on microbial cells. The mechanisms depend on the type of radiation that you're using, but generally radiation either destroys or damages DNA. So you're targeting the replication and the genetic material of the microbe if you're using radiation. So, and then we're looking at surface tension depressants. This is essentially just a very fancy words that describe soap and detergent. So don't feel freaked out by it too much. And they essentially just loosen microbes on surfaces. That's usually what they're used for. And then going on to filtration, I'm sure you're all aware of what that is. This is generally used to treat something that is microbes that are in something that's heat sensitive. So if you have something that you want to preserve that will be destroyed by heat, sterilization is an option that you can consider to remove microbes from that substance without destroying the substance. So if you think of something like antibiotics that are being produced by microbes, you want the antibiotics they produce, not the microbes. But if you were to boil that, you would probably harm the microbial substance or the antibiotic substance. So just to get rid of those microbes, it's much easier to just filter them. Speaking of, this is how that filtration process will then look. So essentially what we have here is a diagram of vacuum filtration. You might have seen these in the labs that with the valve, and then this vacuum line will be plugged into or connected to, is probably a better word, connected to a adapter in the wall that will create suction. And then there is this upper and lower chamber, and because of this vacuum line is creating an area of lower pressure because it's taking out the air, the water, or the substance, I should say, will be pulled down, but between the two chambers, you'll have this membrane filter, and that membrane filter will be too small for the microbes to pass through. Typically, if you want to remove bacteria, you want to use a filter probably or in the vicinity of 0.22 microns in size, because most bacteria tend to be larger than that. Of course, if you're concerned about something like viruses, you want to use a much smaller pore size. So then as you pour your solution through, the microorganisms will be trapped by this filter because they can't pass through. And then the vacuum will pull the vacuum that's been created here by this essentially a low pressure system or low pressure area, will then pull the solution through to the bottom of the chamber, and everything that the filtrate that collects here should then be completely sterile if your filter wasn't compromised in any way. Okay, so some other things that we can look at is osmotic pressure. This is something that you guys should definitely be aware of given our previous lectures. So this works similar to drying, but you don't actually physically dry it. So what do you think I mean by that? Think back to our previous lectures on the movement of water and solutes and how you can manipulate that. So essentially that boils down to how you're using water potential and you're using solutes to manipulate the water potential. So water will always, just as a recap, water will always move from an area of a low solute concentration, so inside the cell, to an area of a high solute concentration, which you will be creating outside the cell. This is why honey is, for example, such a great antimicrobial agent, because 80% sucrose, and few organisms are able to grow in such an environment because they will just not be able to retain water. So far, all the methods that we've discussed are forms of physical control. And with the exception of osmotic pressure that we just discussed, everything else on this slide constitutes forms of chemical agents used to control microorganisms. So we'll be discussing these quickly, and that's essentially starting with gas. So for gas, something such as ethylene oxide or formaldehyde is generally a broad spectrum biocide. The way they work is through alkalization of the carboxyl, hydroxyl, and sulfhydryl groups on the proteins. Looking at alcohols next, they denature proteins and can also disrupt membranes. Heavy metals. So if you look at something like silver, data from silver suggests that these ions denature enzymes. So you are targeting the enzymes of the organism. And they bind these enzymes to reactive groups, resulting in their precipitation and inactivation. So heavy metals typically function or target the enzymes that the microbe needs for its metabolic processes or for reproductive processes. The problem with using heavy metals is, of course, that they are not selective and can bioaccumulate in human and animal cells and have toxic effects. Acids also denature proteins. Nitrate and nitrite can make meat appear red and can, for example, prevent the growth of something like botulism. So that's obviously pretty handy. So we're going to be moving on. I decided to just include this table in case there are still some of you that don't have a textbook. I really want you guys to get on that, especially with the class test coming up. Please make sure you have a textbook. My slides and my discussions and my lectures do go over most of what will be covered in the text or in the test and the exams, but don't be surprised if not all the content comes from the lectures. I do expect you guys to be reading your chapters and to be doing the exercises in the books. I might be pulling some questions from the book, hint, hint, nudge, nudge. So definitely make sure that you guys are looking at that. And this table, I'm not going to be discussing this table, but this is, these slides are a nice way of reviewing what we've just discussed and a nice way of studying for it and preparing for it. So definitely look at these types of tables that summarize the information in a nice, succinct manner. And think of these when you are studying and how I could potentially ask these, because this is such a treasure trove of short form questions. So yeah, just a little hint there, take it or leave it. So now we're going to be looking at chemotherapeutic agents and how they differ from physical and chemical agents in a certain manner. And I would like you to just pause the video here for a second and think if you can figure out how you expect them to be different. So I'm going to tell you now, so if you want to still think on it, pause. But chemotherapy tends to be more selective in its toxicity to microorganisms than the former agents we discussed. So you can target specific microbes or specific cells while leaving others unharmed. So how do we do this? One of the first things that we can do is we can look at something called antibiosis. So antibiosis is simply the biological interaction between two or more organisms that is detrimental to at least one of them. It can also be an antagonistic association between an organism and a metabolic substance produced by another. So here, take home message here, think antibiotics that are produced by certain microbes, which will then bring us to antibiotics and antimicrobials. So what's the difference between an antibiotic and an antimicrobial? Well, the clue is very much in the name here. So first of all, both antibiotics and antimicrobials do the same thing in that they both inhibit the growth of microorganisms. How they differ is in how they are made. Antibiotics are produced by microorganisms, and antimicrobial agents are synthetically made. Many of the antibiotics that we use are actually isolated from soil microbes, interestingly enough. And then we move on to selective toxicity. Remember that we just said that these chemotherapeutic agents are more selective than the previous chemical and physical methods we discussed. And so essentially that boils down to the selective toxicity. So with antibiotics and antimicrobials, they are specific for targeting microbial cells without harming the host cells. Remember, most of these substances are produced by other microbes, so they won't harm all microbes. Differences here between prokaryotic and eukaryotic cells play an important role on the selectivity of these chemotherapeutic agents. So if you look at this picture that we have here, this is a nice example of selective toxicity. There are two organisms growing on this media. There's Penicillium notatum, which is a fungus. You can see it over here. And then this Staph aureus, which is growing here. It's been streaked over here. And this is a bacteria. Notice that there's a clear area between the fungus and the bacteria called the zone of inhibition. And this is produced because Penicillium notatum produces an antibiotic compound called penicillin, which I'm hoping you're aware of, which can interfere with the cell wall formation, and more specifically with the peptidoglycan, which is only found in the bacterial cells. So the fungi will not harm its own cells, and remember, fungi are eukaryotes. They will not harm their own cells because they lack peptidoglycan, but the bacteria will be harmed because they have peptidoglycan. And the substance that's produced by the penicillin that's produced is only harmful because of the peptidoglycan in the bacterial, present in the bacteria. So then there are different modes of action that we can look at, and that we will be discussing here, that these chemotherapeutic agents can implement. They can look to inhibit cell wall synthesis. They can impact cell membrane functions. They can disrupt protein synthesis. They can also disrupt nucleic acid synthesis and functions, and they can also be selected for specific metabolic pathways. So here is an example of how these antimicrobial drugs can function and what they will target. So based on this diagram and the descriptive boxes here, you can see that there are different microbial drugs will target different processes, and I'm going to go through each of them in a little more detail. So, but first we're going to start with this inhibition of synthesis of metabolites, bringing us to sulfamines or sulfa drugs. So here you have what is called competitive inhibition, and that's induced by these antimetabolites. So what is an antimetabolite? An antimetabolite is just a chemical that inhibits the use of a metabolite, pretty explanatory there, which is another chemical that is necessary for metabolism. So sulfamines or sulfa drugs are synthetically made, but obviously we have that over there, and they interfere specifically with the folic acid synthesis, because its structure is similar to para-amino benzoic acid, or benzonic acid, sorry about that, also known as PABA, P-A-B-A. And thus it acts as a competitive inhibitor. Folic acid, just to give you guys the background here, is a coenzyme which is important in the synthesis of purines and pyrimidines, so think DNA. So how does that work exactly? How does what is this competitive inhibition? How does this work? So under normal circumstances, you have your enzyme and your substrate, and your substrate will be bound to your enzyme. Now, what you have with competitive inhibitors is the competitive inhibitor will compete for this site here, will compete with this the normal, the regular substrate for this position here. And if it binds there before the substrate can bind there, the substrate is no longer able to bind there. So just to rephrase that a little, in our normal enzyme and substrate reaction, the substrate will bind on the active site. That's the active site over there, and a product will be produced from that reaction. So this is normal business as usual. Now a competitive inhibitor is introduced, and because of its similar shape to the substrate, it can now bind to this active site of the enzyme, and then it'll prevent the normal reaction from taking place. So the normal reaction is thus inhibited. So in the specific example that we discussed earlier with the PABA, so in a normal reaction, here you have your PABA, it'll bind to dihydro pteroate synthase and form folic acid, this is what happens in this process up here. But when you introduce the sulfur drugs, they disrupt this process, meaning this is never produced. And because these metabolic pathways are essentially a series of reactions, if you shut it down right at the start, the rest of the reaction will not take place, meaning you'll never get to the formation of new DNA and RNA. They won't be able to synthesize, the microbe will not be able to synthesize new DNA or RNA. Just as a note here, because you see the multiple processes, sulfur drugs are often used in combination with this trimethoprim that also block the synthesis of DNA, RNA, and proteins. The way the two drugs work together is a nice example of synergism, and it allows for a broader spectrum of action and reduces the emergence of resistant strains. Next, we'll be looking at the inhibition of cell wall synthesis. This is what something like penicillin does something like this. So antibiotic and antimicrobial drugs that target bacterial cell walls are often considered the magic bullet. And why do you think that is? It's because they do not harm eukaryotic cells, because the eukaryotic cells don't have that structure. The structure differs, so you can use penicillin here and have no fear, or in humans without having any fear that the penicillin will harm the eukaryotic cells of the human. So specifically talking about here about penicillin. So penicillin is actually the most well-known antibiotic, which was accidentally discovered by Alexander Fleming, one of the history's most happy accidents. So essentially his culture was contaminated by a common mold, the Penicillium mold, and he noted this zone of inhibition. Remember when we looked at that slide earlier, it's a bit more visible than it is here, but you can see here where the bacterial colonies are away, are more normal here further away from the fungus, because the fungus is inhibiting the growth. So essentially, even the penicillins we have today still function in the same way. We have altered them a little bit in the sense that we've improved their targeting a bit. So I see we're running a bit out of time, so I'm just trying to move this process along here, and that's why we're going to go now to the two types of natural penicillin, which is G and V. You can see G, which requires an injection, and V, that can only be taken orally. Generally, naturally, penicillin has a narrow spectrum, and they're most effective against only Staphylococci, Streptococci, and some of the spirochetes. And another disadvantage of natural penicillin is the susceptibility to penicillinasis. So remember, the asis just would mean here it's an enzyme that is produced by bacteria that cleaves the B-lactam ring of the penicillin molecule. So semi-synthetic penicillin has been designed to be resistant to this enzyme and to have a broader spectrum. So that's how we've essentially altered the penicillin and improved its use. So here we can see the penicillin that is produced by the Staphylococcus species and the B-lactam ring that is then can interfere with the peptidoglycan crosslinks. Remember, think back to those lectures of how those crosslinks, what they look like in the bacteria. And then, yeah, essentially this is, once this ring can be cleaved open by the penicillinase, and it can change its structure to look like this, meaning it will no longer be effective against the bacteria. This is just by means of an explanation. I do not expect you to know or to draw the what penicillin, the chemical, these diagrams look like. I don't expect that goes beyond what you're expected to know at this point. So now we're going to be looking at the inhibition of protein synthesis. So protein synthesis, just as a bit of background here, is absolutely vital for all cellular functions. So drugs that target protein synthesis will target the 70S ribosomes found in prokaryotic cells. And there are several drugs that target protein synthesis, but we're going to focus specifically on streptomycin. So streptomycin was first produced by Albert Schutz in 1943. And Streptomyces produces streptomycin, which is a member of the actinobacteria, which are gram-positive. This is one of the first antibiotics that is used against gram-negative bacteria, or that can be used against gram-negative bacteria. And it also specifically is effective against Mycobacterium tuberculosis. So if you think back, remember to the Mycobacterium, we discussed them in previous lectures, where they have this unusual waxy coating on its cell wall surface, primarily due to the presence of mycolic acid. And this makes them impervious to gram staining, and as a result, tuberculosis can appear either gram-negative or gram-positive, so it is considered neither gram-positive or gram-negative. And streptomycin is important because they can actually be an effective countermeasure to these mycobacteriums. So how does this work? So streptomycin is a aminoglycoside antibiotic which interferes with the initial step of protein synthesis. We will be going through protein synthesis in later lectures in greater detail, so don't feel too lost at this point. And essentially what this antimicrobial drug does, it changes the shape of the 30S portion of the 70S prokaryotic ribosome, which causes the mRNA to be read incorrectly. So here you can see the 30S portion here, and the streptomycin acts on this portion and essentially causes this code, the DNA, the messenger RNA or mRNA, to be read incorrectly, and thus it will interfere or it'll disrupt the process of protein synthesis. Here you can see some other examples in the sketch of other types of antimicrobial drugs and how they can target this protein synthesis. I do expect you to know the differences between what streptomycin does and what a tetracycline does and what an erythromycin does. I just selected streptomycin as the example today. So just be sure to know these and they're different and how they're different. And I would ask you also to please refer back to the slide and back to this lecture after we have discussed DNA synthesis and RNA, protein synthesis in greater depth, because you will have a much greater understanding of this process, and you will understand that this 30S portion is absolutely vital. So if you are disrupting it through streptomycin, this entire process will essentially fall apart. And then the other action that we can look at is injury to the plasma membrane. So injury to the plasma membrane typically involves changes in its permeability that can lead to losses of important metabolites. So essentially, just as a reminder here, metabolites are important for metabolism. So for example, something like food and energy for growth. And something that the drug that we're going to be discussing here quickly is just polymixin. This is produced by Bacillus polymixa, a gram-positive bacterium. Polymyxins are antibiotics with a general structure consisting of a cyclic peptide and a long hydrophobic tail. They disrupt the structure of the bacterial cell membrane by interacting with the phospholipids. Remember how those phospholipids are arranged. So they are also essentially, if you look at the active ingredients in Neosporin, the first aid antibiotic, that would be a form of this neomycin sulfate, which is in the aminoglycoside family of antibiotics, and they're effective against gram-positive and gram-negative bacteria. And then you look at something like polymyxin B, which we have here, which is a cationic polypeptide antibiotic active against gram-positive and gram-negative bacteria, and they cause injury to the plasma membrane. Then there's also cephalosporin, which is produced by Cephalosporium acremonium, which is a type of fungi, and it works exactly in the same way as penicillin works. So it interrupts the cell wall synthesis. And this is just how those cephalosporins look. I'm sorry for the bit of a rush here. I do have more detail on these slides, but we are running a bit out of time. I do not expect you to know too much of these details, just the fact that what these cephalosporins are and how they function. Please make sure to read those chapters in your books. And here is a nice study or a table that you can refer back to when studying that should provide you with some more details and should ease the studying process a bit, or the preparation. It's not really studying, it's more preparation. And I do expect you to know the sources of these antibiotics, which microbe produces which type of antibiotic. Okay, so now we're going to be looking at antifungal agents, and I want you to just remember back here quickly about why antifungal or why fungi can be harder to treat than bacteria, and that is because they are also eukaryotes. So they can be more tricky because it's hard if they invade a person like a human that also has eukaryotic cells. You have that problem that you want to harm the microbe, but you don't want to harm the host cells. That does create a bit more of a problem at times. Something that can be used is something like nystatin, which is synthesized from Streptomyces nursi. And just in brief, how this affects the fungi, it binds to ergosterol, which is a major component of the fungal cell membrane. So kind of you're targeting again the membrane of the microbe. And when present in sufficient concentrations, it forms pores in the membranes that lead to potassium leakage, acidification, and ultimately the death of the fungus. Importantly, nystatin also impacts the cell membrane potential and the transport by lipid peroxidation. So conjugated double bonds in nystatin structure steal the electron density from ergosterol in the fungal cell membranes. And this lipid peroxidation alters the hydrophobicity of the interior of the channels in the membrane, which are necessary to transport like the ions and the polar molecules. And once again, when you disrupt this transport, and it will result in cell death. Nystatin is an example of something that can be used to treat candida infections, which can include skin infections such as diaper rash, thrush, worse like vaginal yeast infections. Okay, so now looking at an antiviral agent. Here, typically you are trying to target the DNA, so something like ARA works by interfering with the synthesis of viral DNA because it is a nucleoside analog. So this picture over here, you can see that it's been taken underwater, and this is of the Caribbean sponge Tether crypta, from which the nucleosides are isolated. and the nucleosides being, there's no way I'm going to pronounce those names. That's just insane. But these compounds led to the synthesis of a new generation of sugar modified nucleoside analogs and are related to the compounds cytarabine. So a note here, you might have caught there that I used the term nucleoside instead of nucleotide. So I'm not going to go into too much detail here, but a nucleoside, you can be thought of as a nucleotide that lacks a phosphate group. So how this antiviral agent functions is essentially the viral polymerase incorporates the compounds with non-canonical bases, and the compounds are then activated in the cells by being converted into nucleotides. So they are converted from nucleoside to nucleotide. They then block cellular division or viral replication by impairment to the DNA and RNA synthesis, or by inhibition of important cellular or viral enzymes that are involved in the nucleoside-nucleotide metabolism. So here you can see what I was talking about, where they lack the structural similarity here between the nucleoside, the aciclovir, and the deoxyguanosine. So this is just to give you an idea of how similar these things look to each other. Again, I don't expect you to know this for exams. So just to give you a bit more of an idea here of what I just said in that previous slide. So typically your phosphate will bind to your nucleoside, your normal nucleoside, and your host cell enzyme kinase will form, and you will have this guanine nucleotide that will then go through the process of DNA polymerase, or will then be acted upon by DNA polymerase, which is an enzyme, and will be incorporated into the DNA. You'll understand more of this process after our DNA and RNA lectures, which are coming up soon. So essentially how this drug works is it makes sure it gets incorporated into the nucleotide, and then the DNA polymerase will be blocked because it can't incorporate this into the new DNA. So the virus cannot replicate its genetic material anymore. It's essentially being halted. Okay, just be sure to read these chapters. Again, have a look at them in your text. These keep in mind the text pages and might differ between different editions. This is just for my textbook, but I do expect you to have prepared these for exams. We just simply don't have the time to run through them all. So please have a look at them, make sure that you understand them, because they might be appearing on tests or exams. Okay, so what we have here is an example of the disc diffusion test, which you can see here. The clear zones, again, indicate the zones of inhibition, with larger zones indicating a stronger antibiotic response, or a stronger antibiotic. So in this case, size does matter, because this substance, for example, is way more harmful to the organism than this one is. So that's something that you can use to test the efficacy of your antibiotics. Now moving on to what is a major concern currently, and that's antibiotic resistance. So a variety of mutations can lead to antibiotic resistance. And as I mentioned, this is one of the major threats that is currently facing the human race. So essentially what we can have here, what are the mechanisms of antibiotic resistance? You can either have the enzymatic destruction of the drug, where the microbe will simply produce enzymes that can destroy the drug. The microbe can also be altered to become resistant to penetration by the drug. They can also essentially change their target sites, so the drug is not able to find and bind on them anymore. And then essentially these resistance genes are often on the plasmids. I have mentioned this in previous lectures. And the thing with these plasmids are, oh, sorry, I skipped one here. So the rapid ejection of the drug, essentially here is when the drug enters you, you just almost like think of the microbe as spitting it back out immediately. Okay, so that is what the genes will code for. It'll give the microbe like these abilities, or one of these abilities, or a combination of these abilities. And these genes are carried on the plasmids, also known as the transposons, and they can be transferred between the bacteria. So you don't need a replication to occur for these genes to be transferred between the different bacteria. And that's why it becomes such a problem, because if you stop your antibiotic, prescribed antibiotic course early, even if you feel fine, the microbes that you have left in your system can then become resistant to the drug and can flare up again. And now you'll have an infection that's caused by an antibiotic resistant strain. So finish your antibiotics, people. Yeah, this is just a little short that I think was pretty funny, of like how this bacterial transfer, or how the the genes can transfer with this shady microbe, the resistant microbe, essentially giving other microbes the resistance gene, and now this gene will also become resistant. Just a funny little microbe here. So some of the major causes of antibiotic resistance, this is important, obviously for all of you students wanting to go into the medical field, but also just important, I think, because of the nature of this problem and how serious of a problem it is likely to become. Some things that we want to avoid: you want to avoid using outdated or weakened antibiotics. You want to avoid using antibiotics for the common cold or other inappropriate conditions. Reason this is inappropriate is the cold is caused by a virus, not bacteria. So if you're drinking antibiotics, it's not doing anything to the virus. Remember, we've discussed a lot here about how specific chemotherapeutic agents target specific microbes. So if you are drinking antibiotics for a cold, you're doing absolutely nothing to your cold virus. You have to take antivirals. Yes, another thing, big sinner is the use of antibiotics in animal feed, where these animals used for agricultural purposes are just pumped full of these antibiotics, and then it gets expelled in their waste into the environment, and then you will have microbes being exposed to it, but they will also be able to develop resistance against it because they're so they replicate so rapidly, and the few microbes that are present that are resistant will just proliferate, and they will transfer these genes to new microbes. Again, as I said before, failure to complete your prescribed regimen of antibiotics, or using someone else's leftover prescription. This essentially results in the same thing, or the same problem as discussed. We briefly touched earlier on synergism. So synergism essentially is a form of what happens when you combine drugs. And it's essentially when the effect of two of the drugs together is greater than the effect of either of them alone. There's also another term here, antagonism, which you have to be aware of, which is just the opposite. So this is when the effect of the two drugs together is less than the effect of either alone. And here you can see the difference in effect between two drugs, where here this drug is obviously way more effective than this one on this specific microbe. Okay, so here, this is a nice little table to summarize the spectrum of activity of a number of chemotherapeutic drugs. I do want you to know this. So what type of treatment will be effective for what type of bacteria? That penicillin is limited only to gram-positive. Streptomycin is one of the ones that, remember, we discussed the TB and the mycobacteria, and then the gram-negative bacteria. So just go through this, make sure you understand where this comes from and why what goes with what, essentially, what goes where. Okay. And then here, just again, take home message here. Don't keep leftovers of antibiotics for future occasions. They may not work, or you most likely not even have enough to kill all the bacteria. So you will just be contributing to the problem of antibiotic resistance, which, again, in my opinion, this is just an opinion, is one of the major threats facing the human race currently. Okay, so going on to today's review questions. So true or false, fungal infections are often more challenging to treat than bacterial infections. Just going to give you a couple of seconds here. The answer here, of course, is true. Remember, I did review this a couple of times in the slides. Remember that fungi are eukaryotes, so it creates a challenge in terms of targeting the fungal cells that are eukaryotic, but leaving the host's cells unharmed. So true or false, the definition of an antiseptic is an agent that kills or inhibits microbial growth on non-living objects. Okay, I'm going to move on here. Answer here is false. Remember, the devil is in the details here, people. So remember, the difference between a disinfectant and an antiseptic is that a disinfectant is applied to non-living objects and an antiseptic is applied to living tissues. So these details are important, guys. We need to know them. Okay, and this is just going to be a nice little example of a longer question. This is more to just get you guys in the right mindset for exams. Again, this will not be on the test, the class test. Class test will only have short questions. This will be for the longer format questions that you might find in your exams. So state whether you agree or disagree with the following statement and then motivate your answer. The statement reading, a bacteriostatic treatment will compromise the viability of bacteria. Feel free to pause the video if you want to write down your answer or something before you want to check, but I'm going to move on. So your answer here, I'm going to walk you through how I would mark a question like this. So first of all, you have to state whether you agree or disagree. So you have to disagree. If you agree, you're wrong. Disagree, you get 1 mark. Now your motivation. Your answer will have to contain the following information. You have to define what bacteriostasis is, that it's the inhibition of the growth of bacteria without its destruction. And then you also have to tell me what a microbiocide is. What these are treatments that are used to kill microbes, and you can have specific types of So essentially, I want both those definitions. I want the bacteriostasis with the inhibition of bacterial growth without destruction, and then I want you to also tell me which is the one that kills microbes, and then thus you will then tell me that because bacteriostatic treatments do not kill the bacteria, they will not impact the viability. So as a hint here, look at the mark allocations. Typically, I like to mark exams in the form that you will have 1 mark per relevant fact. So for this question, your disagreement will be 1 mark. Your rationale will then provide 4 marks. So 2 for correctly defining bacteriostasis, and then 2 for the microbiocide. And this also tests your insight, because you have to know and understand what bacterial viability means. So yeah, I think that gives you guys some more insight, and I hope you don't, you knock the class test out of the park. And then I will see you guys in the next lecture. Thank you so much. Okay, bye guys.